What is up, guys? In this video I am going to be talking about cancer immunotherapy and chimeric antigen receptor, otherwise known as CAR T cells. And the place that we're going to be working up to with CAR T cells is the most advanced form that we currently have of treating cancers using your body's immune system. And in this video I will also be trying to keep things very high level and use analogies that hopefully you find useful without needing to have too deep of a biology background. And when I think about cancer, I think of bad guys basically running around in your body that we need to kill. And I think of your immune system essentially as a police station or police officers who patrol around your body trying to find the bad guys. And the very earliest forms of cancer immunotherapy are forms in which we basically just give patients lots of stem cells. And stem cells are kind of recruits that have just entered the police force. They have yet to be trained, they have yet to specialize, they can go on to become red blood cells or platelets or a bunch of other immune cells. And what we're doing in these patients is we're essentially giving them immune cells, we're giving them stem cells with the hope that having a stronger immune system will help cure a patient of their cancer. And the problem with this is that cancers are very evasive and very good at getting away from your immune system. And if you saw the movie Blues Brothers in the 1970s, I love the scene with the chase where you've got hundreds of cop cars in Chicago all chasing after the Blues Brothers. And that's kind of what I think about that's going on with this kind of stem cells, is we're basically adding in a lot more immune cells in the hopes that somehow, putting 10 more cop cars onto chasing the Blues Brothers isn't necessarily going to help us catch them any faster. So that's the biggest problem we have with stem cell therapy, but it was an early version that we used in medicine to help our patients out, and that's all we had, so that's what we used. Now, what happened later on is we started getting better at identifying the bad guys. We started having drugs like Rituximab. And when you look at these drug names, I want you to focus on the last two letters. This AB stands for antibody. And essentially, when we think about antibodies, we can think of ways or these proteins that will stick onto the bad guys that will help your immune system recognize them as bad guys. And Rituximab is used specifically for patients who have lymphomas. And in lymphomas, your own immune cells, some of your immune cells, the B cells, are going to have cancer. They're going to be dividing uncontrollably, and this will cause huge problems. And so what Rituximab does is that it will bind onto the bad guys, who in this case are B cells. And this makes it easier for the other immune cells, like the T cells and macrophages or dendritic cells, so all the other kinds of police officers you have in that police station, to identify the bad cops and to go after those bad cops. And that is what Rituximab does, is it just binds onto B cells and it makes them easy to identify, so that if you have essentially B cell cancer, your immune system can recognize the cancerous B cells. And while this does make intuitive sense, and while it can sound like this should really be effective, the issue here is that when we're dealing with patients who have cancer, they're often in very weakened conditions and their immune systems are weak. And so, looking back on just giving patients stem cells, that can make intuitive sense, because if you've got a weakened immune system, if I give you some stem cells, at least your immune system will be stronger and hopefully it can defend itself or find those cancer cells and kill them. But again, we ran into the same problem of we're having a weakened immune system to begin with, and we're basically calling the police station and telling them, here is this bad guy, and that act is the equivalent to binding an antibody onto the bad guys. But in this case, the issue is that the police station or the police officers are just ill-equipped, or they're understaffed, and they can't, even with the help of us saying, here's the bad guy, it still cannot fight itself off. And this does buy patients some time, or sometimes maybe this is very effective, but it still had some room for improvement. And with Rituximab, we're basically calling 911 to help the police identify who the bad guys are. Now, what else can we do besides just identify bad guys? Well, in addition to identifying the bad guys, another drug came along called Blinatumomab. It's right here. It came from Amgen. And what Blinatumomab does, and again, notice how we've got the AB ending. So again, it's an antibody that helps us identify the bad guys. But what Blinatumomab has in addition to the bad guy identifying property is something called a CD3 receptor. And this CD3 receptor was identified by scientists that will essentially alert and attract T cells into the area where they're needed most. And CD3 receptors, don't get too focused on all these numbers and letters. Basically, you can think of it like a van that would take a SWAT team to where they're needed. And that's how I think of it. So basically, we are calling 911 and we are providing your immune system with means by which it can get the right immune cells where they're needed and identify who the bad guys are at the scene. And so we're going a step further in immunotherapy in helping your immune system not only recognize the bad guys, but also get to the scene of the crime so they can fight off the cancer. Now, again, unfortunately with cancer, it is such a terrible disease because we're dealing with weakened immune systems. And even though I can get my T cells right where they're needed and tell them exactly who to kill, or yeah, basically that's what it is, it still isn't enough because your immune system, especially with oncology, we're generally in damage control and we've already lost so many police officers that just getting them there still isn't enough for patients. It's a step forward. It helps us buy people more time, but it still isn't enough. And so what we are currently doing and what is currently being developed is something called CAR T cell therapy. And CAR T cell therapy involves us literally taking out your T cells from your body, these T killer cells, and training them to identify the specific cancer that you as a patient have. So we would take a biopsy of your cancer, we would take your T cells, we would train your T cells and give them chimeric antigen receptors, which are these little red things you see in this diagram, in order to help them have a really high affinity for the bad guys. We would make tons of these CAR T cells and we would give them back to the patient. Essentially what we're doing here, sticking to my analogy, is we are training mercenaries. We are training people, we are training these T cells to identify the specific cancer that this patient has so that we can really help your immune system. And in vitro means in a test tube outside of the patient, we're able to add in resources and energy to create tons of CAR T cells that we can give back to the patient so that the patient has an immune system that we have helped really push along and have not only the ability to identify the bad guys and be there at the scene of the crime, but also have the weapons that they need in order to kill the bad guys, your cancer cells. And the biggest challenge we currently face with CAR T cell therapy is the cost of development. And oncology and the price of medications is astronomically high. It costs many tens, hundreds of thousands of dollars to develop CAR T cell therapy for patients because a lot of this stuff is still in R&D. And why is it in R&D and why can't we just move forward with this stuff? It's because so much engineering and development and quality control needs to go in to make sure that these CAR T cells that we give to patients have effective tools and they will not kill the patient themselves, because a lot of this again is just damage control. But, and this is seen in the early generations of the CAR T cells that we used, the CAR receptor that was engineered had too high of an affinity for bad guy cells, and this caused the immune response to be so strong that it ended up killing the patients. And so as we continue to do our R&D, as we continue to have better quality, we are identifying ways to manufacture CAR T cells that are much more effective at recognizing bad guys, not killing good guys, and killing the bad guys. And so this is a very exciting field and time in science, and I hope you guys have found this video useful for CAR T cell therapy and cancer immunotherapy. Thank you so much for watching. Let me know if you have any questions, and take care.